Hello. Welcome to Spotlight on Science from St George's University of London. Today is Monday the 30th of March. My name is Julian Ma and I'd like to welcome you to this second video we've made about coronavirus. At the moment, while we're all stuck at home, exposed to the news 24 hours a day, it's very easy to get quite down about the prospects of coronavirus and how long it will take us to get out of this current situation. And the purpose of this video is to give you some reasons for hope, looking around the world at the developments that are going on in not just the number of cases that are being reported, but also developments in the areas of medical research. Later on in the video, I'm going to be introducing you to three of my colleagues, Professor Tariq Sadiq, who will be telling us about the latest developments in diagnostics for coronavirus, Dr. Elisabetta Groppelli, who will be telling us about the treatments that are being used for coronavirus and the exploration of new medicines to treat coronavirus in hospitals. And then Professor Paul Heath, who will be telling us about the latest advances in developing a vaccine against coronavirus. I'd like to start, though, by just taking a look at what's happening in Asia at the moment and looking at some of the good news stories that are coming out. Of course, we all know that coronavirus started in a city called Wuhan in the province of Hubei in China. And whilst they experienced enormous numbers of cases and fatalities, if you look at this graph showing the total coronavirus cases in China, you can see that this has plateaued at around 80,000. And in fact, the latest data coming out from China shows that there has been almost no new cases of coronavirus in China for some days now. And this is shown in the bottom graph. Hubei, of course, was the epicenter of the pandemic, with 67,000 confirmed cases, 3,000 deaths, but about 62,000 people recovered from the disease. And the situation in Hubei has got to the point now that the authorities have deemed it safe to start letting people move around the province again, especially to travel into the province. I think this is good news for everybody because it shows that given the right measures and the right compliance by the population, that there is a chance to control the outbreak. It's not just in Hubei that we have seen this kind of pattern. In South Korea, with a similar population to Hubei province, they managed to keep their numbers of deaths relatively low at only 152. And although the number of new cases is still climbing, you can see that they are not climbing at that exponential rate that we're seeing in Europe and in other parts of the world. They have managed to control the virus, and the number of new cases per day in South Korea, like in China, has dropped dramatically and is now at a relatively low level. I think this gives hope for everybody that provided we take the scientific advice and we follow the scientific advice, that we will be able to get our coronavirus outbreak in the UK also under control. I think the other important thing to recognise is that the maths support this. And I've taken this very nice graphic from the BBC, which shows what happens when one person passes on the infection to others in circumstances where there is no restriction around social contact compared to under the isolation circumstances we find ourselves in now. Different viruses have different levels of infectivity. And the coronavirus has what's called an R0 of 2.5. That means, on average, given no restrictions, every one person with the virus will pass it on to 2.5 other people. And with no restrictions on social contact, within 30 days, every individual with the virus who passes on to 2.5 people, and those 2.5 people pass it on to 2.5 people each, we would expect 406 people to become infected with the virus. Now, with our social distancing measures and our isolation measures in place, the hope is that we can at least halve this infectivity. If we can get it down to 1.25, then you can see the impact after 30 days. Instead of infecting 406 people per original individual, only 15 people would become infected. That's a dramatic drop, and that's exactly how we are going to manage to bring down the new cases of coronaviruses in the UK. The point about this, though, of course, is everybody has to comply with this. Every person who doesn't comply risks infecting that 406 people in 30 days. And so for this to work, for the maths to come to reality, we all need to social distance and isolate ourselves. I'd like to introduce Professor Tariq Sadiq, who is a Professor of Molecular Medicine at St George's. He's also a consultant in HIV and sexual health medicine. Thanks for joining us, Tariq. And
to be here.
I wanted to ask you about the latest developments in the development of diagnostics, but perhaps the first question really is that we already know how to diagnose COVID-19 or coronavirus infection. So can you tell us why it's important to develop new diagnostics?
Well, we do know how to diagnose it with the laboratory tests that we have, but we're not completely sure about how accurate those tests are. So it's quite important to continue the development, the testing, and the evaluation, even of the diagnostics that we have, because this illness is a very new illness, and so we don't have all the information that we need to be able to say confidently that the tests work perfectly. And that's important because these things normally take a bit of time, but the urgent need for a response has meant that a number of tests have just had to be adopted very quickly so that we can do something.
Okay.
And so it's quite important to continue to develop not just tests, but increase the accuracy of the tests and probably the speed of the tests for those reasons.
And why is speed important here?
Well, speed is important so that we can do a number of things. So, for example, if you've got somebody who you want to isolate as quickly as possible and get a real diagnosis, then having to wait even for a day or two can be a problem. But if you're able to diagnose them quickly, you might be able to trace their contacts more quickly. You may be able to implement containment measures around them more readily. But it's also important not just for containing patients who might be infecting and isolating them, but it's also important to allow key workers, for example, and other members of the public to return to work if they haven't got.
Yes,
and that's really key. I mean, in the NHS, I mean, one of the defining things about this epidemic is that it's not just about people being ill, it's about the NHS system being overwhelmed and healthcare workers becoming unwell with relatively minor illnesses, but which prevent them from taking part in care. So enabling people to safely return to work in key areas of work and of the economy is really quite important. So having rapid tests that can give you results in minutes rather than a day or two, and making sure that they're accurate is really quite important.
So as well as the diagnostic test, we're also hearing about something called an antibody test. So can you tell us what the difference is, please?
An antibody test is not quite telling you whether you've got the infection right now. It's telling you that you've had the infection at some point in the past. And that may still be now, but it may be that you've had it and it's resolved. So it's a way of just letting doctors, but also public health professionals, know how much infection has been around. We're not quite sure how long that antibody response lasts for, but certainly in the short, medium term, it's going to be there. So we can get a sense of the population, how much of the infection is already in the population. It's also something that might be useful to let people know whether they've been being infected in the past and now might be safe to go to work.
Yes.
Symptoms.
Yeah, I can see how that's important. So what are the What are the sort of new technologies that are being applied to diagnostics that will enable us to do things faster?
Well, I mean, the key thing is that these lateral flow tests might be useful.
What's the lateral flow test?
Sorry, so the lateral flow tests are these antibody and these antigen tests, tests that are similar to pregnancy tests.
Okay.
So you put a tiny amount of sample, maybe a swab sample from a patient from their nose, and then stick it onto a tiny little stick, and the sample will just migrate across the material, and you'll get two lines, just like a pregnancy test. And they can detect two things: antibody, which is your body's immune response to tell you whether you've had the infection in the past, or antigen, which is actually the real virus, and the presence of the virus indicates real infection.
And like a pregnancy
types of tests that we can do that are quick.
Yeah, I was going to say, like a pregnancy test, that would be really quick, right?
That would be quick. The difficulty with the tests, these kinds of tests, and we're evaluating these tests at St George's, is that just historically they tend to be less accurate for many infections. Not for all, but for many infections, they tend not to be as good as the other tests that we're doing in the laboratory that are called molecular tests. Those are much more accurate because they look for DNA, or in this case RNA, the gene of the virus, but use a method called PCR, which is just amplifies the amount of those genes that are present.
So tell us a bit more about the molecular tests. What can you do there?
So with the molecular tests, they can be used in the laboratory, and the advantage of doing it in the laboratory, even though it might take a little bit longer because it's got to be done in the laboratory, it's got to be turned around in terms of running the test. They tend to be large machines. You've got to make sure the results are right, and then batch testing. They're quite good. They can be quite accurate. You can get to very low levels of the virus that you can detect in the sample. So they're quite good that way. But again, speed is an issue. But there is similar kinds of tests that have now been developed for a number of years, and incredibly have been immediately deployed for the detection of COVID since the outbreak, that are very, very rapid. And there's a good there was a test, I think it was all in the news a couple of days ago, that Abbott, the company Abbott, have produced a molecular test that detects the SARS-CoV-2 virus, the virus that causes COVID-19, within 5 minutes.
Oh, fantastic.
That's amazing. 5 minutes to detect it, and then 30 minutes to rule it out. That's often how it works.
In the last few days, we've been hearing more about whole genome sequencing and using those technologies. How will that help?
So, actually, when whole genome sequencing was used initially on the virus, that was immediately helpful, because whole genome sequencing was the method that he actually discovered the virus. It's actually quite novel to find infections in this way, but over the last few years, people have been doing what we call random sequencing or unbiased sequencing, and that's enabled scientists in China, in the Hubei province, Wuhan, to actually just look for everything that might be there in the samples. And by using that unbiased process, whole genome sequencing has allowed us to actually detect a brand new coronavirus. And it was found in all the samples who had the same kind of illnesses. That essentially allowed us to discover the cause very, very quickly and confirmed with other experiments. There's another use of whole genome sequencing. The virus actually doesn't change that much over time. It's relatively well conserved. Its genes don't change in the way that many other viruses and many other infections do. But it changes just enough to be able to detect where these viruses are spreading and how they're spreading.
So you can track them.
So tracking and surveillance, I think, is another really important part of the role of whole genome sequencing.
Okay, well, thank you very much, Tariq. That was really interesting, and I do wish you all the best with the development of your assays. I know you're working hard on that. Thank you.
Thank you very much. Thank you.
So now I'd like to introduce Dr. Elisabetta Groppelli. Elisabetta is the lecturer in global health at St George's, and thank you for joining us, Elisabetta.
It's a pleasure.
I'd like to talk to you about treatments for COVID-19, please. And actually, just before we start, I think one thing that's on everyone's minds is, what to treat themselves with at home. And the general advice has been to use paracetamol. And there's just been this issue about ibuprofen. And I was wondering if you can just enlighten us on that now.
I think it's a really interesting situation that has been developed, not necessarily from scientific evidence, but we've seen how the conversation can be influenced by, high-level political figures, for example, all over the world. And the ibuprofen is one case. So the unfortunate situation at the moment is that there isn't a consensus when it comes to how to treat COVID-19, in any areas of medicines. And therefore, this leaves us open to potential observations that might actually need to be put into a context. So the how to take this information about ibuprofen. So the WHO and our own Public Health England have obviously made the decision of playing it on the safe side. So they have said we do recognize that there has been report of ibuprofen being detrimental to the outcome of the progression of COVID-19, and therefore we do support the use of paracetamol, which can do similar things in this context. Of course, with the caveat that if patients already are on ibuprofen, they need to check with their own physicians about continuing the treatment or not.
Okay, thank you very much. So actually here I'm a little bit more interested in talking about the treatments which are being developed for COVID-19 when you have to go into hospital and when the disease gets a bit more serious. And I understand at the moment we don't really have any specific treatments which are standardized.
Absolutely. This is the unfortunate situation of not only when a new virus comes to the scene, to the stage, but also generally speaking, we don't have that many antivirals that are really usable in these situations. So this means that treatment in hospital is very much a supportive care. So, however, we can, looking at the virus and looking at what we've learned so far in the only past few months, there's been some intelligence, let's call it this way, some information about the virus that actually supported what we call the repurposing of drugs. This means that there are drugs out there already used in the clinic, either for some viruses or other diseases, that do make sense if we use them against COVID-19. And this is what's been done now all over the world, including the UK.
Okay. So can you give us an idea of some of the more promising candidates?
So the most promising candidate has been looked at is a drug that was developed during the Ebola outbreak. This is only a few years ago. And then in vitro studies, when they looked at the action of the drug against the virus, also took into consideration other viruses, including MERS and SARS, the first one. And they've seen some effect. The unfortunate thing about Ebola is that drug wasn't effective against Ebola. But that obviously meant that we had some information about the coronaviruses. So that was the prompt to use that drug. How does this drug work? This drug inhibits a particular enzyme of the virus. Basically, as we know, the virus needs to go into a human and to a cell and then start replicating. And it needs to make lots of copies of the genome where the genetic information is stored. And there is an enzyme called RNA-dependent RNA polymerase. But basically, all it does is putting all the bits on a string together and to making then the new copies of the genome. And this drug actually blocks this chain reaction, and therefore the virus doesn't make the genome, and therefore the virus doesn't go on to infecting other people. So this is great. The drug is called remdesivir. Apologies, it's a bit of a mouthful, but that's what it is, and it has been trialed now for COVID-19.
That sounds very promising.
And are there any other drugs which are in development at the moment that looks good?
Yes, there's been drugs that are used in the clinic for HIV. So these are part of the antiretroviral therapeutics. And these act in a different way because they inhibit other enzymes, other active proteins of the virus. And these are proteases. Basically, I can say when my grandmother taught me how to make gnocchi. You make a long little sausage of pasta, and then you chop it into bits. So the proteases are the enzymes that chop the sausage into the active building blocks. And so having a drug that inhibits this process means that, of course, you want the little gnocchis, for your pasta dish. You don't want the entire sausage. And the reason why it is possible to look at HIV drug is because, it's almost like these proteins are like scissors. They do have a handle that might be made of different materials, of different colors, and that's specific to the virus. However, the blades are the same. And so this drug will actually inhibit the blades, and therefore it could work for different viruses, even if the viruses are different.
I've also heard about a malaria drug that is being used that might be useful in COVID-19. Can you tell us anything about that?
Yeah, so the malaria drug you're referring to is called chloroquine, and there is a version of it, sort of like a second version, which is called hydroxychloroquine, and it was developed just to decrease the toxicity of the drug, so kind of like an improvement. From the molecular point of view, actually, I found this drug very interesting because this is a drug that we use in vitro, which means when we look at the virus in cells, to specifically inhibit a step of the cell, which is acidification of the endosome. Don't worry, this simply means that it goes on to attack and change a particular part of the cell that the virus might require. And so we use this drug to see if this particular acidification is required for lots of other viruses. However, we cannot really explain how this actually seemed to be working in humans in the context of a body, in the context of a patient. And it would be really interesting to start to pinpoint and dissecting this. There is something to be said, however, about chloroquine. And although it's a really, it's an old drug, and it's a good old drug, it's been along for quite some time and it's doing its job. But it seems that the dose required to actually have an effect on the COVID-19 patients might be towards the higher limit, which means that toxicity, side effect might actually be a big problem. So at the moment, I think some clinical trials are going on with chloroquine, but is the other types of drugs, including antiretrovirals and the RNA polymerases that are gaining more traction.
Okay. And with these new drug candidates, what is needed to get them into generalized use? I mean, how do we go from a drug that we think might work to a drug that actually we can use on the clinic?
The short answer is very much we need to do clinical trials. So there is a rationale, there is approvals, and then the reality is that we need to put the drugs into trials and see if they have an effect or not. There are different complexities when it comes to clinical trials, but I think it's very important to point out that all the authorities, led by WHO at the moment, are really simplifying all the paperwork that needs to be done to fast track the use of these drugs, obviously in safety, but with efficacy specifically as the outcome. And of course, when it comes to the lab, so away from patients, we are still looking at developing new drugs and coming up with different scissors inhibitors or different different, beats on the strings inhibitors. That, however, will require longer time. But again, we do have an immediate action, and we also have a longer term agenda, and both of them need to be pursued.
And I suppose the advantage of using these repurposed drugs that you mentioned is that we already know quite a bit about the fact that they might be safe in patients anyway. And now for those drugs, it's just a question of do they work or not in COVID-19?
Yes, the rationale definitely takes that into consideration, but also takes into consideration the fact that maybe a different dose might be required. Okay. For example, if the drug was specifically developed against HIV, it might work against another virus, but maybe a higher dose might be required. And in that case, then again, in the clinical setting, it will be important not to look at efficacy, but also safety, because, you cannot just up the dose until it works. The patients needs to be put first.
Okay, well, thank you. It really sounds like there's a lot of research going on and clinical activity as well, which is exciting. So, Elisabetta, given your sort of best guess, when do you think the first we will see of these drugs might be? How soon might that happen?
So, one thing that is both good and bad, I think, is that unfortunately at the moment there is a really high number of patients, which means that actually we do have the opportunity of gathering data quite quickly. Mm-hmm. Obviously, numbers still need to be collated, and there are worldwide trials. So it's still a matter of months. So I think it would be, when we think about how long should we expect ourselves to be in this pandemic situation, this supports the fact that it's still going to be a few months before we have weapons that are on top of the social distancing that are so crucial now. But it would be great if we get in a few months something more therapeutic.
Sure. I mean, whilst we're desperate for these drugs, the important thing is they don't do more harm than actually not using them, right?
Absolutely.
Elisabetta, thank you very much for joining us, and I wish you all the best of luck with your research. Thank you.
Thank you.
I'd like to introduce you now to Professor Paul Heath, who is a professor and consultant in paediatric infectious diseases at St George's University of London and St George's Hospital. Hi there, Paul. How are you doing?
Hi, Julian. I'm well, thank you.
Paul, I'm happy that you've joined us because I'm hoping that you'll be able to tell us about what's the latest advances in vaccines against coronavirus. This is one thing I guess we're all anxious to hear about.
Indeed. Absolutely, Julian. This clearly is a field that is exploding at the moment, and there is much interest in the development of a vaccine against this virus that is now causing the global pandemic. And I say this is a very active field. In fact, there's a website that is currently monitoring all of the research activity around COVID-19, and currently there are 46 different vaccines in different stages of development against this virus. So this is important and encouraging, but we are still some way from being able to implement an effective vaccine against the virus. And much of the effort also began really with the development of vaccines against the other important coronaviruses that have caused severe disease. So the SARS coronavirus and the MERS coronavirus. And so there is quite a lot that has been learnt already based on vaccines that have been developed against those viruses.
And so how is it thought that these vaccines might work?
We have to go back, I think, to the pathogen that we're dealing with, which is, of course, an RNA virus. And this virus, we know a lot about how it causes disease. And from the point of view of vaccines, one of the really important aspects and one of the main focus of many of the vaccines that are being developed is the way the virus engages with human cells. Mm-hmm. And it does so by binding to a receptor in the host cell. There are four main structural proteins for this virus, mm-hmm. and the one about which there's been most focus with regard to vaccine development is the spike or S protein, and that is because it is important for modulating viral entry and membrane fusion, a critical part of the pathogenesis of the disease. And so the spike protein, either the whole length of the spike protein or components of that spike protein, of which there's a number, but the S1 and S2 components, the S1 for binding and the S2 for fusion of the virus, have been the focus of, as I say, many of the vaccines that are under development.
Mm-hmm. So can you tell us what is typically required to get a vaccine, let's say, from the laboratory into the hospital?
So the human demonstration of the vaccine goes through 3 or 4 different phases, the phase 1, phase 2, phase 3, and then the phase 4 trial. Mm. And in brief, phase 1 trial is a small number of subjects, particularly looking at safety. Mm. The phase 2 is a larger number, looking at safety, but also looking at the immune responses. Mm-hmm. Phase 3 is larger trial, which typically is to demonstrate whether this vaccine is effective at preventing the disease in question. And then phase 4 is a post-implementation in a very large number in the field, as it were. Mm-hmm. Once the vaccine is licensed, it's implemented, and then effectiveness and safety with large numbers can be assessed.
So to get through the phase 1, 2, and 3 trials sounds like a lot of work and might take quite a lot of time. How will things be different for coronavirus?
Well, I think the process will be the same. I expect that the speed of these trials will be much faster, because clearly there is a huge need for a vaccine against this virus. And then the process of moving from phase 1 to phase 2 to phase 3 will be as rapid as possible. And of course, that's really important. But it's also important, of course, that the safety of the vaccine is characterized fully, and that there are no shortcuts taken. And of course, there won't be. Mm-hmm. There will be tremendous enthusiasm in progressing this vaccine. Enthusiasm, I think, from clinicians, from researchers who will work together, so generate and collect large numbers of subjects. And I suspect there will be enthusiasm from people in the UK and elsewhere in participating in such trials too. So I think a collected effort will mean a much more rapid progress through those phases of vaccine development than perhaps we have typically seen.
So the vaccine you mentioned in the UK, is that the one from Oxford?
Yes, that's right, Julian. So this is a adenovirus vectored vaccine in which the adenovirus, harmless in humans, is the way of presenting the virus protein from the SARS coronavirus 2. And this is now about to go into phase 1 human trials in the UK.
And that approach of modifying the adenovirus, I think they've used before for other infectious diseases, haven't they?
Yes, they have. They have used it for a number of other important infections, including TB, malaria, Ebola, and perhaps most relevant to this virus, for the MERS virus, the Middle East Respiratory Syndrome virus, which is, of course, another coronavirus. So similar technology. And indeed, therefore, has been used in a large number. This technology has been used in a large number of humans, but there isn't as yet a licensed vaccine using this technology.
Okay, but promising nonetheless, because there's all that background information from the other diseases.
An extremely promising approach, and so I think we are, of course, very pleased that it is now entering phase 1 human trials in the UK, and look forward to the rapid progress of these trials, which we and others hope to participate in.
So finally, perhaps, I mean, given a favourable wind, how long do you think it might be before any of these vaccines make it to the clinic?
Well, of course, there are so many hurdles to be passed through before we get there. I think the optimistic thinking around this is of the order of 18 months from now. Mm-hmm. That sort of speed. Of course, I think what we have is such a huge global collaborative effort that perhaps we will do even better than we have done in progressing vaccines than in the past. Mm-hmm. So it will be even shorter than that. But that, I think, is a generally accepted ballpark before we can implement it. And then the question will be, then there are many questions about implementation. Who will be the first people that groups in which we implement it? Will it go into younger adults, into children, healthcare workers? What about pregnant women? What about the elderly, where the disease is so important? What about the immunocompromised? And all of these things will, of course, depend on which vaccine it is that we use, because some vaccines will be more appropriate, for example, for those with weakened immune systems than others. And then it will also depend on where we are at that point in terms of, for example, herd immunity. A lot of work needs to be done in understanding what will be the optimal time to vaccinate.
And given that this is a global pandemic, is there thinking around the accessibility of these vaccines for people in lower and middle income countries?
There is, and of course there has to be, because, as you rightly point out, this is a global problem, and there is great fear about how this disease will affect people in low-income countries. It's not clear yet. I think it's fair to say how important the burden is there, but it is clear that typically they receive vaccines, or they have access to vaccines much later than we do in high-income countries. But I think that is foremost in the thinking for many vaccine developers and those that fund them.
Thank you. Well, thank you very much, Paul. It's been really fascinating to hear about all those developments. Thanks for joining us.
Thank you, Julian.
Well, thanks to my colleagues who have given us such useful information about latest developments in coronavirus research. I'd also like to thank my son Thomas, who, with me in isolation at home, has helped put this video together. As I have said in the past, the coronavirus situation changes very quickly, and it is important to keep up to date using reliable sources. The NHS, of course, continues to provide the best website, telling you what to do if you suspect that you have symptoms or whether other people in your household have become ill. If you're interested in following the global pandemic, the World Health Organization also has an exceptionally good website to follow and to keep a track on latest developments. If you're interested in finding out about that new vaccine that has been developed in Oxford, the website for the Oxford Vaccine Group is also shown on this slide. Finally, for those of you who might have developed an appetite for learning about medical research and are fed up of coronavirus, we do have a number of Spotlight events which we videoed and put online. These are slightly longer than your normal TED Talks, about an hour each. But you can see on this slide that if you search for Spotlight on Science and St George's, you will find a number of fascinating videos. On the right here is a list of all the topics that we've been covering over the last year, for example. Well, thank you for joining us. I hope this has been a useful video and that you have learned something new. For now, stay safe and stay at home.